Welcome to our today's podcast, and we are your host.
So today we are diving into the fascinating world of biotechnology, the science where biology meets technology to shape the future of life itself. From decoding DNAs to designing the crops that feed millions of people, biotechnology is transforming how we live, how we heal, and how we form the shape of our future.
So grab your curiosity and let's uncover the biotechnological stories.
Well, myself Megha.
I am Nishikant. Welcome to today's podcast, where we will learn about protoplast, isolation, and fusion, the heart of somatic hybridization.
So imagine taking two different plants, a juicy tomato and a hearty potato, and combining them together into a new plant. It sounds scientific fiction, right? But that's what protoplast isolation and fusion allows us to do in the lab. So the first step is selection and preparation of the plant material. We will select a healthy and a young plant tissue
Why young plant leaves?
because their cell walls are thin and easy to digest with the enzymes. Then the next steps come, the surface sterilization is done using ethanol. So we will cut the plant tissue into small strips and place it into the osmoticum solution. Okay, so now comes the most fantastic part, that is the cell wall digestion, means the removal of the cell wall. So we will immerse the leaf tissue into a solution containing cellulose and pectinase, which will dissolve the middle lamella of the leaf. And after a few hours of the gentle shaking, cells will lose their cell walls and become the protoplast. Now comes the next step, which is the filtration and centrifuge. We will separate the newly formed protoplast from the cell debris, filter the mixture through the nylon mesh or centrifuge it at the low speed and discard the supernatant. We can also check the viability of the newly formed protoplast using a stain known as FDA stain, which will stain the viable protoplast into green colors.
Okay. Now I will explain the further process. Now comes the magic moment, that is protoplast fusion. We can make two protoplast fuse by two different methods. First one is chemical method, in which we use PEG, polyethylene glycol, and second method is electrical fusion using electric pulse.
Okay, so can you now tell me that which method is most commonly used?
Electrical fusion is most commonly used because it is more precise than chemical fusion.
Oh.
After fusion, we don't only get hybrid production, but some protoplasts remain unfused.
Oh, then?
Then so we select fused ones and culture them in media. Over time, these protoplasts will regenerate, the cells wall starts dividing and form callus. So, do you know what is callus?
Yes, I know. Callus is a mass of unstructured and unorganized cells.
Finally, the callus is transferred to regeneration medium and subculturing is done to trigger their shoot and root formation, and then there you have it, a newly formed hybrid.
Oh wow, that's wonderful.
So then they are acclimatized and tested to confirm that they are true hybrid or not.
For example?
We can take example of tomato, hybrid of potato and tomato.
So that's all for today. Today we have learned each and every step of protoplast isolation and fusion, from selecting the X plant to regenerating into a whole new plant. And we can also say that somatic hybridization helps the scientists to overcome the sexual incompatibility barriers by combining the useful traits at the cellular level.
So now here is question for you guys. If you could fuse any two plants to create your own hybrid, Which one would you choose and why?
Share your answer. So thank you for tuning into the episode. Stay curious and innovative.
Okay,
Bye.
bye.